Okay, so welcome to this next video in which we are discussing the nicotinic acetylcholine receptors. Okay, so we've now discussed the neuromuscular junction form of the nicotinic acetylcholine receptor, both the adult version and the fetal version. We've also discussed certain drugs which interact with the neuromuscular junction nicotinic acetylcholine receptor. We are now going to talk about the ganglionic form of the nicotinic acetylcholine receptor, which is in the autonomic ganglia. So let me just give you a brief reminder of what the autonomic ganglia are, and then we'll look at the structure, the subunit composition of the ganglionic nicotinic acetylcholine receptors. Okay, so basically, in the autonomic nervous system, you have two divisions of the autonomic nervous system, the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system is involved in fight or flight responses. So when you're in a situation where you need to either run or fight, then basically you're going to activate the sympathetic nervous system, and it's going to cause loads of different systems to alter their function so that you're ready for a fight or flight response. So basically, when you are really, really terrified, the things that are happening to you, that's maybe, partially at least, a responses that are produced by the sympathetic nervous system. In contrast, the parasympathetic nervous system is often called the rest and digest nervous system. So it's active when you're relaxed and calm, and it causes physiological responses that are relevant for you being relaxed and calm, such as digestion. Okay, right. So, the neurons that are involved in the autonomic nervous system, they come out of the spinal cord. So let's draw the spinal cord here. So here, this is the spinal cord, okay, like so. So this is the spinal cord. Well, and basically, the neurons that are involved in the autonomic nervous system will come out of the spinal cord. But the question is, do they instantly do they directly synapse onto their target? And the answer is no, in most cases anyway. There is one major exception, which is the there is one major exception, which is the adrenal gland. But in most cases, the autonomic nerve which comes out of the spinal cord does not synapse directly on its target. Instead, what happens is it synapses on another neuron, which then synapses on the target. Okay, so let's say this is the effector, is the fancy word for target. Okay, right. So, this first neuron that has its cell body in the spinal cord and then synapses onto a second neuron, this is known as the pre-ganglionic neuron. So this is the let me bring this over a little bit, pre-ganglionic neuron. Okay? And the second neuron in this pathway, which synapses onto the effector here, this is known as the post-ganglionic neuron. Okay, right. And this synapse between the two, basically what happens is you don't just have these in isolation. Instead, what happens is that you have loads of these all happening at once. So basically you'll have another pre-ganglionic neuron coming out here, and it will synapse onto its post-ganglionic neuron at the same place, basically. And this other post-ganglionic neuron might have a very different target, basically. So let's say it's got a more something that looks more round, like that. So a very different target. But basically, all of this synapsing is occurring in one portion of the body, in one little site. And this place where you have all these synapses, this is known as an autonomic ganglia. So you have sympathetic and parasympathetic ganglia, where sympathetic pre-ganglionic neurons synapse onto their post-ganglionic neurons, and where parasympathetic pre-ganglionic neurons synapse onto their post-ganglionic neurons. So basically, these autonomic ganglia, as they're called, They only contain either sympathetic neurons or parasympathetic neurons. They don't contain both. And it's where all the cell bodies of the post-ganglionic neurons are stored, basically. Okay, now, the neurotransmitter which is used between the pre-ganglionic neuron and the post-ganglionic neuron here is acetylcholine. Okay, so that means we need an acetylcholine receptor on our post-ganglionic neuron, and it is a nicotinic acetylcholine receptor that you have on these post-ganglionic neurons. And this form of the nicotinic acetylcholine receptor is different. It has a different subunit composition to the neuromuscular junction nicotinic acetylcholine receptors that we've seen. Okay, so the form of the nicotinic acetylcholine receptor that is found predominantly, and you know, if you search hard enough, you'll be able to find articles where it tells you about other forms of the nicotinic acetylcholine receptor in the autonomic ganglia. But the main one, the predominant one, is known as the ganglionic nicotinic acetylcholine receptor. Okay, and it's got the subunit composition alpha 3. 2, so you have 2 alpha 3 subunits, and then also beta 4, and then you have 3 lots of those. Okay, right, so let me show you in our cartwheel diagram the structure of this nicotinic acetylcholine receptor. So again, it's a pentamer of these subunits, basically, and we now want to see how these different subunits are arranged. Okay, so remember there were these 17 different subunits, basically, that the human genome had, and we saw that there was alpha 1 to alpha 10, beta 1 to beta 4, then there was the gamma, delta, and epsilon. So this contains only 2 of those. It contains alpha 3 subunits. It needs 2 of those to build the receptor, and it needs 3 beta 4 subunits. So let me show you how they're arranged relative to one another. Okay, so here's our pentamer again. And basically what you have is you'll have the alpha 3 subunits here. Okay, so let me just think how I'm going to do this. We'll want the alpha 3 subunit here. Okay, I'm just thinking about where I need to write it so that I can show you the A, B, C, D, E, F loops again. Okay, and then we have beta 4 subunits in the other 3 positions. So let me colour this in now. Right, so we'll have alpha 3, we'll have alpha 3 in blue here. So I'll outline the alpha 3 subunits in blue. So you take the alpha 3 nicotinic acetylcholine receptor gene, you make 2 of those proteins, okay? Then you take the beta 4 nicotinic acetylcholine receptor gene, and you make 3 of those proteins, and you stick these 2 alpha 3 proteins together with these 3 beta 4 proteins, and that creates you an alpha 3 2 beta 4 3 heteropentamer. So again, it's a heteropentamer because you've used different nicotinic acetylcholine receptor subunits in your 5 slots, basically. You haven't used the same one 5 times. Okay, right. So let me now show you the binding sites for acetylcholine on this alpha 3 2 beta 4 3 heteropentamer. Okay, so basically, these alpha 3 subunits, they act as the principal subunit again. So they are the ones which are going to provide the A, B, C subunits. The A, B, C loops. Okay, so these are the principal subunits. So they're going to provide these A, B, C loops in their protein structure. So here's the A loop, here's the B loop, and here's the C loop. Okay, so do I have room to label them up? Maybe. There's an A, there's B, there's C. Okay, in this beta 4, this then acts as the complementary subunit. So these 2 here are going to act as the complementary subunit. Okay, and in fact I'll just add in the A, B, C's onto this one here as well. So here's A, here's B, here's C. Okay, and now this beta 4 and this beta 4 are going to provide the C, D, and E. The D, E, and F loops, although we know they're not loops, they're beta-pleated sheets, which are going to form the other half, the acetylcholine binding site. So these are the complementary subunits. Okay, so here comes the D loop, or the beta-pleated sheet. Here's the E loop, and here's the F loop. And again, here's the D loop, the E loop, and the F loop on those two beta-4 subunits, which are basically next to the A, B, C domains of the alpha-3 subunits, which they are neighbouring. Okay, so again, this heteropentameric nicotinic acetylcholine receptor, which we find in the autonomic ganglia on the post-ganglionic neurons, it has two binding sites for acetylcholine. And we'll see that this is going to be different when we look at the alpha-7 homopentamer, which is in the central nervous system. Okay, so two acetylcholine molecules bind to this nicotinic acetylcholine receptor. One here and one here. And again, the binding sites are between two subunits. So, two acetylcholine molecules come and bind. It causes this receptor to open. It will then allow sodium ions to move into the cell, depolarizing the cell, causing an excitatory postsynaptic potential, which will then increase the chance that the postganglionic neuron will fire. Okay, now let's discuss pharmacological agonists and antagonists. Okay, right, so the endogenous agonist is obviously acetylcholine. So acetylcholine will come, bind to those binding sites, and will increase the likelihood that the receptor will be open. Okay, a non-endogenous agonist is nicotine. So nicotine is an agonist for this alpha-3-2 beta-4-3 heteropentameric nicotinic acetylcholine receptor. So two nicotine molecules will come and bind here, one here and one here, and they will activate this receptor. Okay, and that's going to cause increased activity in these postganglionic neurons, whether they are sympathetic or parasympathetic neurons. And that's quite odd, because you're going to get both an increase in activity of the parasympathetic nervous system and an increase in the activity of the sympathetic nervous system. Okay, right. And now let's discuss an antagonist for these receptors. And this is a specific antagonist which is selective to this form of the nicotinic acetylcholine receptor. This doesn't interact with any of the other types that we're going to see in these videos. So it doesn't interact with either the fetal or the adult skeletal muscle nicotinic acetylcholine receptor, and it doesn't interact with either of the brain forms that we're going to see in the next video. Okay, and this is the drug trimetafusp, also known as trimethafusp. So I'll write those two names, trimetafusp, or you might also hear this referred to as trimethafusp. So it's like indomethacin and indomethacin. Okay, yes. So trimetafusp and trimethafusp, they're both names for the same drug, basically. And this drug is a competitive antagonist. So all it does is it binds to these acetylcholine binding sites here and will stop the acetylcholine from being able to bind. So if you take trimetafusp, what it will do is it will bind to these alpha-3-2 beta-4-3 heteropentramers on the postganglionic neurons in the autonomic ganglia, and it will stop acetylcholine from being able to bind. So when acetylcholine is released by the pre-ganglionic autonomic neurons, then basically it won't be able to have any effect on the postganglionic neuron. So you're going to stop the function of both the sympathetic and the parasympathetic neurons. And I want to stress that even though the neurotransmitters that the postganglionic neurons release are very different in the case of the sympathetic and the parasympathetic nervous system, the neurotransmitter that is released from the pre-ganglionic neuron onto the post-ganglionic neuron is the same whether this pre-ganglionic neuron is a sympathetic neuron or a parasympathetic neuron. So it's acetylcholine for both the sympathetic nervous system, which I'll write as SNS, sympathetic nervous system, and the parasympathetic nervous system, the PNS. So trimetaphlon or trimethaphlon will block that ganglionic neurotransmission in both the sympathetic nervous system and the parasympathetic nervous system. Okay, so that's the ganglionic nicotinic acetylcholine receptor. In the next video, what we'll do is we'll move on to the brain isoforms, starting with the alpha-7 homopentamer.